Hello, my name's Dr. Martin Khechara and I'm from the University of Wolverhampton and from the fantastic STEM response team. Over the next week, we're going to be doing some catch-up sessions to do with biology for those of you who might be going into A-levels or going into BTEC and further education. So today, I just want to give you an idea of what we're going to be covering, but also start to give you some of the information that you might need to go into your studies later on. And we're going to talk about biological molecules today. So this is what we're going to be covering in our biology catch-up sessions. We're going to do biological molecules, and I'm going to do that with you now. We're going to be looking at cellular structures and plant biology. We're going to be looking at genetics and cell division, the circulatory system and the exchange systems of the body. We're going to be looking at variation, classification and evolution. And we're going to be looking at some maths that's essential for your subject area in biology. So if you want to know more about the STEM response team and find our fantastic resources, you can find us on all these social media channels, and also you can see our web address there at the University of Wolverhampton, where all these fantastic videos are going to be hosted. So let's consider some important biological molecules. Really, there are only 4 groups of important biological molecules that we need to consider at this stage. Firstly, carbohydrates, lipids, proteins and enzymes, and nucleic acids. And I'm going to touch on each of these briefly as we go through the content today. So carbohydrates then, let's start there. So they are important, of course, because they are sources of energy for our body. They help to provide life for respiration, to help us live. Of course, these are the sugars and things like starch and cellulose. And actually sugars, particularly like glucose, that we're going to be talking about in a minute, are absolutely essential because it's what your brain runs on. So let's start with sugars then. Well, these are small water-soluble molecules, and of course, they taste sweet. There's 2 different kinds really we need to talk about now, and they are the monosaccharides, things like glucose, which I've already mentioned, fructose that you find in fruit, and galactose. But if we start to combine them using a clever bit of chemistry, called a condensation reaction, what we can actually do is we can make disaccharides, like the sucrose that you put maybe in your cup of tea or your coffee. That is really just a disaccharide that consists of fructose and glucose. And when we join the 2 together, we get a little bit of water made. That's why it's called a condensation reaction. Important to know as well that glucose actually exists in 2 forms, depending on where, as you can see in the diagram here on the slide next to me, where the actual hydroxyl group is, this OH group. It depends actually where it is, whether it's at the top or at the bottom. If it's at the bottom, it's alpha glucose, and if it's at the top, this is called beta glucose. So we might not think that starch is a carbohydrate, but actually what it is a really big polymer made of glucose, and it's actually made of polysaccharides called amylose and amylopectin. And what these are, they're actually polymers of glucose. So we have a polymer made of polymers, and that giant polysaccharide, which is starch, it's got loads of branches, and because of that it all sticks together in a big sort of globule and becomes insoluble, and it actually allows plants to store energy. And of course we can break that down using something I'm going to talk about later on called an enzyme to actually provide energy for our body, because we can break it down from the starch into smaller and smaller and smaller molecules until we can get to the glucose to feed our brains. If you think about plants and plant cell walls, we're going to talk about that later in other revision sessions. We can look at cellulose, and this is a polysaccharide that's only found in plant cells. Now, it's not like starch in that it's all messy and all branched and insoluble in that sort of way. It's more organised because the binding between the glucose monomers, because again, this is a polysaccharide, it's a polymer of glucose, the binding actually makes nice long strands. Now, those strands can bunch together to form what we call microfibrils. So we actually get lots and lots and lots of them bunching together in sort of like ropes, and it forms a very strong structure. Now the diagram's a little bit small. You can see these glucose molecules all in a line there on the slide next to me. And if you look very closely, there's some dotted lines. Now, they're dotted lines are something called hydrogen bonds. And actually, they are holding all of these molecules together, and that force is very weak. But that's multiplied and multiplied and multiplied over a fantastically large molecule once that polymer's all put together. So it all holds these strands together. Okay, so moving on to lipids then, and again, these are essential because they form essential components of the cells of your body. And actually, all they are, all right, are fatty acids with a molecule of glycerol. And commonly, they have 3 fatty acid legs with a bit of glycerol, right? And that's called a triglyceride. And they are very, very, very common, particularly in animal cells. And so if we consider fatty acids for a minute then, all they are very, very long chains of carbon, oxygen and hydrogen, quite simply. But at the end, there's something called a carboxylic acid group, and that's this COOH group that you can see there on the slide. Now, we might hear about saturated or unsaturated fatty acids. Now, it's very easy. Saturated fatty acids have just got single bonds all down their massive long chain. Now, unsaturated fatty acids have occasionally a double bond. Now, if they've got lots and lots and lots of double bonds, we could say they are polyunsaturated. So again, it depends on the kind and amount of bonds in that molecule to its sort of chemical characteristics. A really, really important kind of lipid is something called a phospholipid. Now, it is an essential component of animal cell membranes, and it's got some very, very peculiar properties. It's got your glycerol, and this time it hasn't got 3 fatty acids, it's got 2 fatty acid sort of legs, if you like, and you can see on the diagram there. But what it's got is a phosphate group, sort of tagged on the top, and that changes the chemical properties of the ends of this molecule, because the end with the phosphate group is a water-liking chemical group, and we call that hydrophilic. On the fatty acid chains, we have a water-hating group, and they are called hydrophobic. And that means that all the hydrophobic ends sort of move away from the sort of wet environment of the inside of an animal cell. So what they do is they naturally form a bilayer, and that naturally forms into a sphere. And that is how cells are actually formed. And we have this animal cell membrane model made out of this sort of like bilayer of these phospholipid molecules, and it's absolutely essential for life. Okay, so let's move on to proteins then in our little tour through biological macromolecules here. And proteins are made primarily out of amino acids. Now, there are 20 amino acids that are possible, and they come together in different orders to make proteins. Now, you can see a diagram of a typical amino acid there, and you can see there's a blue part with some H's on. That's called an amino group. And we've got a grey carbon with 2 red oxygens and a little hydrogen on there, and that's the acid group. So it's the amino acid. Now, that R group is the only thing that makes the 20 amino acids different, and whatever's plugged onto that R bit changes the amino acids' chemical and sort of structural and charge properties. Now, you can imagine really all the different sort of combinations of all those 20 amino acids, and the order in which they're in changes their shape later on and actually changes the way they function. So proteins can have a number of different sort of arrangements, if you like. They have a primary structure, which is just like beads on a necklace. It's just the amino acids just all plugged in together. They have a secondary structure, and often these are in what we call coils or helices, or sort of zigzaggy sheets, and we call those pleated sheets or beta pleated sheets. We see a tertiary structure, and that's when a protein starts to fold and those different amino acids interact in a molecular sort of way, and they start to bond together through ionic and sulfide and hydrogen bonds. And that actually forms a sort of big blob, a globular structure. Then we start to get quite cool things happening. If we get more than one protein coming together to form a sort of super protein globule, if you like, we say that's got quaternary structure. And many of the things in biology do have this quaternary structure because these larger proteins can actually do a job, as you'll see. Because a lot of these proteins with quaternary structure are what we call enzymes. I'm going to look at that in a sec. Just to sort of summarise then, you can see this diagram with primary, secondary, tertiary and our quaternary structure there with an explanation of each of them. So I mentioned enzymes, and these are absolutely essential to life. They allow the chemistry of biological life to happen at the temperatures that it does. 37 degrees isn't really that warm, and things, you know, chemistry can't happen very quickly there. Any sort of reaction needs something called activation energy. I mean, if we light a Bunsen burner in class, we still have to put a match or a spill to that Bunsen burner to actually get that gas to light. That's because the oxidation reaction that's happening to make that fire occur needs activation energy. We need to put energy into the system. Now, if we can lower that activation energy, the actual process, the chemistry, can happen at a lower temperature. And that's exactly what enzymes do. They are biological catalysts. And they're often involved in things like metabolism, and they're involved in breaking down molecules. A really important one that not everybody has, interestingly, is amylase. Now, if you eat bread, if you chew a little piece of bread, you'll notice if you keep chewing it, it gets sweeter and sweeter and sweeter. And that's because the enzyme, this big protein machine, is actually breaking down the starch in the bread to glucose, like we saw earlier, to give you a sort of sweet taste. So you can actually see and feel it happening. Now, enzymes work with a substrate, and that substrate is the thing that is changed. Now, enzymes have a particular shape, and they have a place on them called an active site. So if you see on our diagram here, the enzymes, they're sort of blue part, and the substrate, which is the thing that's going to be changed by the chemical process that the enzyme is going to help happen, is the substrate, okay, is the sort of purple part. And actually, the place where it binds on the enzyme, the substrate, is very specific indeed. Indeed, most enzymes only actually catalyse one particular enzymatic process. And actually, they're quite sensitive, and they've evolved to actually work at 37 degrees in the body, and those that are used in the body in particular, because if we increase the temperature to a certain extent, they will get quicker and quicker, and eventually it'll get too hot, and all the sort of bonds, the chemical bonds that are holding that sort of globule together and the active site together break down, and that protein undergoes what we call denaturation. We can see this actually when we cook a fried egg. When you cook an egg in a frying pan, the protein that's in there in the white of an egg, called albumin, will denature with temperature and turn white because it becomes insoluble and non-functional, a bit like how an enzyme would stop working with increasing temperature. If we increase the amount of free hydrogen, protons, increase the pH, for instance, or decrease the pH, then actually we start to see a similar effect as the chemistry that holds all of these protein molecules together starts to break down. One thing I do want to mention, I'm not going to mention it too much detail because it's going to be picked up in another session that the team's going to do, is nucleotides and the larger molecule these make, which of course is deoxyribose nucleic acid, or DNA. Now really, nucleotides are just sugars. There's 4 different kinds. There's adenine, cytosine, guanine and thymine, and these make up the very program of life itself. As you'll find out, there are other variations on a theme which are similar to DNA, which encode different kinds of information that allow the proteins we've just been talking about to actually be made in the human body. So just remember, there are 4 main kinds of biological molecules that we need to consider. They are the carbohydrates, the lipids, proteins, and nucleotides. Well, I hope you found that useful, and we hope you enjoy the rest of our revision content that the STEM Response team is going to be putting out over the next month. Thank you.